Hello guys, my name is Hanisha. As a part of my assignment, today I am going to tell you about plasmid vectors. Plasmid is a type of vector commonly used in the recombinant DNA technology. Plasmid vectors are small circular DNA molecules that are used to transfer genetic material between different organisms. Most plasmid vectors are artificially created in the laboratory. They have higher replication rate and are widely used in the field of genetic engineering and biotechnology. Structure of plasmid vector. Plasmids are the small circular or linearly double-stranded DNA molecule. Plasmid vector typically contain an origin of replication, selectable markers, and a multiple cloning site. The origin of replication allows the plasmid to replicate independently of the host cell's chromosomal DNA. Selectable markers are the gene that confers resistance to antibiotics or other substances allow the researchers to select for cells that have taken up the plasmids. One of the important site in plasmid vectors is multiple cloning site. It is also known as polylinker. The multiple cloning site is a short DNA sequence containing several EQ restriction enzyme recognition site. These sites allows researchers to insert DNA fragment into the plasmid vector at specific locations. The multi-cloning site is often located within a reporter gene, which allows researchers to easily screen for successful insertion of the DNA fragments. These sites allows researchers to insert DNA fragments into the plasmid vector at specific locations. Types of plasmid vector. There are several types of plasmid vectors, including expression vector, cloning vectors, and sequencing vectors. Here we are discussing about the expression vectors and cloning vectors. Expression vectors are used to design to produce high levels of recombinant protein in host cell, whereas cloning vectors are used to amplify and purify DNA fragments for further analysis. Differences between cloning vector and expression vector. Cloning vector is used to acquire multiple copies of the foreign DNA fragments, whereas expression vector is utilized to acquire the gene which might be RNA or protein of the DNA. Application of plasmid vectors. Scientists use plasmids extensively as vector for cloning, manipulating, and transferring genes. Plasmids has been key to the development of molecular biotechnology. They act as delivery vehicle or vector to introducing foreign DNA into the bacteria. Using plasmids for DNA delivery begins in the 1970s when DNA from other organisms was first cut and pasted into the species site within the plasmid DNA. Plasmid vectors has numerous applications in molecular biology and biotechnology. They are used to study gene function, produce recombinant protein, and create genetically modified organs. Plasmid vectors can be used in the gene therapy to deliver therapeutic gene to patients with genetic diseases. Advantage of plasmid vector. Plasmid vectors are easy to manipulate and can be customized for specific applications. It can be isolated easily because of small size, more stable because of circular centrifugation, replicated independently of the host cell, high copy numbers, detection easily because of antibiotic resistance gene. They are inexpensive and can be produced in large quantity. Plasmid vectors can be transferred between different organs, allow for the creation of transgenic organs. And here we're discussing about the limitations of plasmid vectors. Plasmid vectors can only carry relatively small DNA fragments. They also be unstable and prone to mutations or deletions. Some host cells may not be able to take up plasmid vectors, limiting their usefulness. To overcome these limitations, scientists has came with many new vectors such as phases, cosmids, back and ag. And here we are seeing some safety concerns about the plasmid vectors. Plasmid vectors may contain antibiotic resistant gene that can be potentially spread to other bacteria and create antibiotic resistance gene. They may also contain oncogenes that could potentially cause cancer in transgenic organisms. Strict safety protocol and regulations are in place to prevent the accidental release of genetically modified organisms into the environment. Some of the future directions. Advantages in genetic engineering and synthetic biology are likely to lead to the development of the newly and more powerful plasmid vectors. These vectors may be designed to carry out large DNA fragments or to target specific tissues or cell types. Plasmid vectors may also be used in the combination with other gene edition techniques, such as Cas9, to create precise genetic modifications. Conclusion about plasmid vectors. Plasmid vectors are essential tool in molecular biology and biotechnology research. They have numerous applications and advantages, but also carry safety concerns. Advantages in genetic engineering and synthetic biology are likely to lead to the development of new and more powerful plasmid vectors in the future. Thank you.